Hello, everybody. Welcome to the Cancer and Evolution Q&A session with Heng. We appreciate you coming here. I'm Tana Marshall. I'm Perry Marshall's daughter. I'm just here to keep things peaceful and running smoothly, so you probably won't hear much from me. Henry, take it away.
Okay, can you hear me? Oh, hi.
Hi.
Hi, everyone. Hi, Dennis. Hi, Carlos.
How
Rock,
are you, Harry?
and Jim. Yeah, I cannot see you. So,
to listen to your talk.
okay. So let me first use just a few minutes, just briefly, recall a little bit about the, let me see. So I need to share this.
Just let me know if you have any trouble, Henry, and I'll.
I actually try to. Oh, hi, Dr. Weinberg. So anyway, so I try to share some briefly reviews a few points. So the first point we actually discuss is we realize that the cancer evolution can be divided into two phases. So the first one is rather dramatic, is also all the genome reshuffling, genome chaos. So after that, so the change the system, the phase transition, after finish that, they enter to the second phase of stepwise Darwinian phase. So they can stochastically, with the help of different oncogene, to grow the population. So basically the two incidents. One is create new system. Secondly is increase the quantity of the system. So that is the first thing we discussed. And in order to understand this phenomena, I mean, for the cancer, you always have the different transition, multiple transition. One is immortalization, for example, the transformation, and then you have become invasive, and finally drug resistant. So each one is a huge population transition. So that is a really phenotype change for us. But then we've actually realized every time this happens, the genome type also drastically change. So we said maybe we need thinking about why the chromosome is so dominant. So to understand this, we actually realize the chromosome themselves, specifically the karyotype, is a new coding. Basically, what they're doing is provide the physical platform for these different genes or different sequences to interact with each other to form the network. So basically, chromosome is not just the carrier of the gene, but is the organizer, right? So therefore, we realized above the gene, the information, you have another layer of information to control the biological system. So this is rather important because in the past 100 years, people always thinking is the gene information control the phenotype, even though they have interaction of the environment. But nowadays, because of the missing inheritability, and because it's so difficult to link the gene information to the phenotype, and we think it's important to consider the higher level of information. So this is the second part. And the third part, if there is a new chromosomal coding information, how such information can be preserved? Because evolution is all about when you have new information, you preserve such information. So that's the basis of our thinking how the biology should understand. So we actually try to search for the mechanism of keep the karyotype information. And it turns out is goes through the function of sexual reproduction, because the meiosis is chromosome pairing. So when the chromosome pairing, two chromosomes just checking the order of the gene. So if it's the same order, you can go ahead. But it's a different order, you'll be aborted in such a way. So by thinking that way, we also found that the developmental program becomes fundamental. Why? Because initial development separate the germline and the somatic cell. So the germline can be working by the meiosis later on to make the system do not change. And meanwhile, by separate of the somatic cell and the germline, the somatic cell could have as much change as possible. So therefore, karyotype can change, copy number can change, gene mutation can change. So therefore, the somatic world themselves have the function for adaptation. And yet, as long as this individual passes the genome, the species will be there, right? So now, I mean, understand this is very, very striking, because for over 130 years, we always thinking the function of sexual reproduction is provide the diversity, because mother or father different, you mix the gene, then you have different combinations. So that's all the idea. But why we have such an idea for so long is because after Darwin proposed the small change variable, people try to find this variable. So when people say, oh, meiosis chromosome can mix this and provide the variable, everyone is on the tree. Say, oh, it's great if you have, so that's the idea. But sometimes this is actually striking, because I studied meiosis for a couple of years. So under microscope, the crossover, the frequency is extremely low, right? So you cannot find so many. If just for the sake of producer diversity, you should see tons of the crossover. But actually, the number is rather limited. And the second point is, the evolution after environmental interaction, they finally have combination. Why you want to immediately break up such a combination? Doesn't make sense. And finally, if the chromosome meiosis just for the sake of mixed gene for the diversity, then if you study the distribution of the sexual reproduction species and the asexual, you should anticipate in the very harsh environment, you should always have sexual, because if they give many, many change. But the fact is opposite. In the harsh environment, you always find asexual species. So this actually completely, challenges this idea. So that's why we actually gradually, go through this one. And finally, because this key evolutionary problem for function of sex can be challenged, how about evolutionary theory themselves, right? So that's a logical step. So we actually realized that from cancer research, the microevolution and the macroevolution is not as simple as we have been told. We were thinking from very beginning, Darwinian idea is, you have a small change, but must be small, but if you're big, therefore you're out of the game, right? So you get small and gradually accumulate for many, many generations, and finally you become another species. But during cancer research, especially during drug resistant, we put the drug there, in few hours, they either survived or be killed. They don't have time for many, many generations to accumulate the survivalness, right? So not just that, when we put the drug treatment, they immediately induce massive genome reorganization. So it's not like previously we thought, oh, you already have some lucky guy or unlucky guy hiding there, and then you kill the sensitive one, the resistant one. take over. No, it's not. It's a completely human treatment-induced monster, overwhelming in such a system. So based on all of those, we actually realized that maybe the cancer is not a typical neo-Darwinian evolution. It's not you accumulate a gene mutation in 1, 2, 3, and gradually you have a big hit. So this is fundamentally different. So that's basically what I have told you in the last week's seminar. So after the seminar, people actually ask a couple of questions, but I will later on, I mean, during you ask questions, I can read back to you about it. And meanwhile, I want to open the floor to everyone, so to ask any question, and especially some critical point if I missed. I most appreciate this type of question because will keep us on track. Because when you propose some of idea, you don't want to yourself miss some important facts. So that's what I really appreciate. So now from here, I will open the discussion or question to everyone else. So Rafi want to ask a question. Yeah, go ahead.
What does Genome Chaos suggest about the proper approach to treating cancer?
So for the cancer treatment for a long time, we always try to kill the cancer cell. So that's a chemotherapy. And the targeted specific therapy also, try to, if you have particular gene mutation profile, so you can kill them, which that's the idea. But the drug resistant always come back. And also people gradually realized, either by clinical observation and the computer simulation, the more effective the killing, actually the comeback is quicker. Now this is a paradoxical as such thinking. So after realize the treatment itself could induce the genome chaos, we actually realized the treatment themselves, for many cases, is extremely harmful. Why? Because we, in the name of kill the cancer cell, we actually help cancer quickly go through the macroevolution and become moving target, become dominant. So therefore, we really need somehow to avoid such a drastically nuclear weapon approach. But because we realize the cancer are two phases, one is the macroevolution, one is the microevolution. So the microevolution, the gene mutation makes the population become bigger. So we suggest that in the future, we can use method not directly target the cancer cell. If we want to kill them, we use a more moderate way to just reduce the size, but still maintain the cancer cell in certain degree under the constraint. So not allow them to become crazy. That is a very good strategy, because in that way, so we always thinking this may be the way of the adaptive therapy. So if you use moderate treatment and make the constrain the cancer cell, but not completely wipe them out. If you completely wipe them out, the new system will emerge to occupy that space to fight with you. So those actually is one of very important one. And the second point is, we suggest sometimes build the constraint of the higher order, like non-specific immuno strain and some other strain, social strain, the even psychology or the family care, this strain, lifestyle strain, not direct target the cancer cell, but constrain the speed, right? So that is another idea to answer your question. Yeah.
Excellent.
Thanks.
Dennis Noble would like to say, great work and ideas. I like the experiments and concepts that lead to the genome chaos idea. And then the question is, but should we stop at the karyotype, excuse me? Should we stop at the karyotype? I would think that all soma characteristics might influence what is inherited.
Should we What should we stop? Let me see. Oh, okay. So this is difficult to answer the question, Dennis. So the every system, every cell has their own phenotype. So we cannot just simply stop the phenotype, because if some people actually joking say that if you really want to cure cancer, you kill that patient, right? You kill the cancer in such a sense. So for the chemotherapy for us, we are at the disadvantage stage, because if you kill this cancer cell very effectively, and the chances are the normal cell will become abnormal, right? Under stress. So that actually is extremely difficult. So if you use very, very powerful way to kill this cell, even if you can eliminate all the cancer cell, then you cause the system chaos. The normal cell also will become crazy as such. So that is a paradoxical approach for us. We have to understand this. This is one point. Second point, the reason the cell karyotype change at the first place, some people is an accident, like a chemical accident, exposure or something. But most people is because of the cellular adaptation. Because as I just told you, so the evolution have two fundamental functions. One is change, because environmental change, you have to change individual. But evolution have another function which have been ignored since Darwin, is actually species is not change, right? So that's the two things, two places. So you change, but you do not change. So the do not change is germinal meiosis, and you just repair very effectively under the karyotype coding, so you do not change. Therefore, somatic cell, you have to be able to change. Otherwise, you cannot fulfill biological function. For example, for liver, we always give this example. For the liver, newborn, human, 46 chromosome, mouse, 40 chromosome, beautiful. But as soon as the mice and the humans start to eating, they become aneuploid, because they cannot handle, because every genome have limitation. If they want to handle, they need to change, right? So at the age of 40, 50, 70% of human liver is completely polyploidy. It's not the human cell anymore. So if we, get serious to thinking about this question. But with all this change, what is the trade-off? The trade-off is they could become out of control of the system, and they emerge the cancer. So therefore, the why me question of cancer is the price we pay for the evolution, right? So that is the situation. So therefore we are not be able to wipe out the change the karyotype. If you wiped out some other population will immediately jump in. Just almost like in US, we always fight terrorism. We always kill them, and then the new group is pumped up. Why? It's because that's the system behavior. When the system is unstable, some place is going to pump it out as such. So, but by constraint, I think it should be very effective. Another thing is right now, human lifestyle changed dramatically. We fly to Europe, we cross all of this. All of this, we need to pay the price as a system, right? So that's actually is interesting. So someone actually, as Dennis suggested, someone said, okay, we should target the genome instability, we solve the issue. But actually, I thinking, for some cancer like the leukemia, so it's the whole system. Maybe if you can replace the bone marrow, you kill them. So that actually could work, as Dennis suggested. This is replaced. But for the solid tumor, we don't know where they are, so it's extremely difficult to replace them at this moment. So I always thinking to constrain will be a better way, right? Anyway, that's my personal opinion. Yeah.
Well, Dennis had a comment rather than a question, which was, Balance between change and no change leads to Darwin's idea of period of rapid change and periods of stasis, an idea resurrected by Eldridge and Gould. This supports your approach.
So, okay, this is a very interesting question. I'd like to give a little bit more background. So, for the Darwinian initially noticed sometimes have the spots, quickly change somewhat in the snow. But this observation is not of his so-called framework of thinking, because if you read Darwin, every time he emphasizes the change must be small, must be accumulation from longer time. Because initially, when Darwin actually he himself to synthesize this idea, he realized it's difficult to tell people, because there's no evidence in nature just showing gradually they become new species. So therefore, he said if he can use the artificial selection as an example, then people will bridge this knowledge, right? So that's this idea. So from Darwin's point of view, the artificial selection is so effective, right? Like the British, they have lots of dogs, they have pigeons, they have many, many ways. But after we really thinking about the cancer, because in the cancer situation, we realized the punctuated evolution and the stepwise evolution is different mechanism. One is driven by the chromosome reshuffling, another is by the gene mutation and epigenetic. So therefore, this scale different. Let us realize that it's not that simply small accumulation, bang, you become big one. So, but if this is not the case, why Darwin have believed this so much, right? We just dig out Darwin's thought. As it turns out, the key idea is artificial selection. You can make all these things happen. You can make the big, huge dog, you can make a small dog. But when we compare artificial selection and the nature selection, we realized it's fundamentally different, cannot even comparable, because we always thinking must be true. Why cannot be comparable? Because artificial selection, we have desires mind in it. So we always use the same criteria, which is push the feature, not push the reproduction, not push the survival. We just give every food to them, we just push the feature like, but the nature always select reproduction and the survival. It's not the feature, it's not the tail of the, the roast, right? But we artificial selection, we totally change the game. This is one. Secondly, in many, many generations, we just focus on same criteria to push this selection. But the nature is totally different. The nature always swing back and forth. So after 100 years, go this direction, you accumulated something, and another 100 years wiped out completely into different direction, right? So that's why when the Peter Grant, the family, the say, sometimes the pig become, the Darwin's finch become big, immediately change the weather, it becomes small. So after all, right, even though 30 years, you see the big, small change, but after a few hundred years, they did not change. Almost like the fashion design. You can say this year different from next year, but from 1,000 years, you didn't see the change. That's exactly like a horse, like anything people analyzed, exactly like that situation. So back to Jago. Jago, they found the very important, they found the fossil, they have drastic and then gradually, what they found is a phenotype. Is a phenotype of the species. So this is extremely important discovery, but he was very confused later on. The reason is he still believed small accumulation become big change. So where is this, why this fossil in the middle suddenly so big change? He cannot solve this puzzle, so therefore he, assuming that the species go to other places to finish the transformation, they come back to this place, right? Which doesn't make sense either. So that's why 6-7 years before he passed away, he wrote the evolution, construction of evolution, the big book. He clearly said, "Oh, when I was young, we found the punctual evolution. No, actually, that's no big deal because there's just a time scale difference. Now we know it's not a time scale difference. What we have found is we found the phenotype change. We also found the genome type change corresponding to the phenotype change. We know it's all of a sudden become new species rather than gradually accumulate. So it's a fundamentally different concept." Yeah.
All right. Well, Rafi said, so it sounds like the actual origins of new species is not Darwinian, gradual/microevolution, but rather macroevolution induced by massive stress to the species at the population level. Is that correct?
Most of them, yes, I agree with that. But then we actually realized that after species already formed, you need a microevolution to help the population grow. So that's the thing, right? But we are not sure how important it is for this phenomenon. But we know, for example, if in the isolated geography situation, for example, in the mountain top, so if people never come back, they always stay in the top. So the beta globin could become the gene frequency could become accumulate after thousands of years. That is true. But for most of the species in Amazon, in every place, they can move around. So that will be never happen, right? So now we know that Darwin in the small island found the dramatically change all of the species, which also not happen in the vast majority of situation, right? So now we know, for example, in the fish live in the cave, right? So they lost the eyesight. So they develop something new feature, even bone change. But if they come out, people cross those species with a different cage. One generation, they bring back all the eyesight. They bring back everything. So the feature and who they are, the species, is totally different concept. So if the chromosome do not change, you are same species. Despite you could have different feature, you could have eye, you could have, but that is not important. So the important is how to define the system themselves rather than the feature. So that's actually very drastically different from traditional thinking. Traditional thinking always thinking is you gradually change in many generations, then you become new species. Now we're thinking, no, just according to cancer, the idea is one generation father already produced daughter completely different. But then you need a few months to make this cell become many copy, right? So that is the idea of the microevolution. So.
Perfect, thank you. So we have another, actually we have 3 questions from Paul Blois, I don't know, sorry Paul, don't know how to pronounce your last name. The first question is, why is the Kindle version of your book so expensive? Was the price set by the publisher?
Yes, exactly. So because the academic, so I always argue with them. I said, I want people read the book, right? But it turns out for the academic, all this book, what they are knowing, the owner can sell very limited. Most of the library buy it. So they want to put a high price. So even just a few library buy them, they're not losing money. So that's all the stuff. So I just, debate with them to say how, they said, okay, if you really want a book cheap, you go ahead to write the bestseller if you can. I'm not winning in this one. So I really want more people to read, to ask them to cut the price, but that's the way it is. But then they said, nowadays, if you want to borrow a chapter or even an article online, you need to pay $30, right? So then in that case, your book is not expensive. So I said, okay, I'm not.
Keep fighting the good fight, Henry. That's why we're putting all of these recordings on YouTube for free. And these things should be available to the general public, at least for a small fee. So question number 2 from Paul is, do you think genome chaos, major gene reshuffling, happens beforehand, causing cancer, these polyploid cells described by James Shapiro and Kenneth Pienta, in the first place? If so, in what ways? Like through x-rays and the feeling of stress, etc.?
So the stress actually is quite interesting thing in the cell from cancer perspective, because the thing is without stress we even don't have cancer. Of course, without stress we even don't have life, as such talking about. So from the immortalization model, what we have done in the in vitro, and then we did the mouse tissue, I continually culture every time to see this, what's the stress. So in fact, it's fascinating is during the cellular growth or whatever, so the heterogeneity themselves could be fundamentally stressed, right? So even you don't have anything else, the life themselves is produces stress, because without the stress, T cell even cannot differentiation. So we know we have to lead the stress, but you also need to pay the price. So that's the thing I cannot, I do not know how to solve the philosophical conflict with such. For example, the mitochondria produce energy, they also produce the stress, right? So that's the thing. So you have to have this one. So therefore, another question related to this one is predisposable, the gene mutation, for example. For example, the BRCA1, BRCA2, breast cancer, this gene. People are always thinking, how unfortunately for this individual have such a gene mutation. But we know during experiment, so if you have p53 mutation for most normal people, then you have more chance to get a cancer. But p53 mutation could rescue these people have BRCA1 or BRCA2 mutation, right? So if without p53 mutation, you cannot born. But with p53 mutation, you actually can have a life. You can produce your children, and then you got the cancer. So which option is better? We actually need thinking more deeply about this issue. Another thing is the p53 mutation, right? Even though for people, unfortunately have this one, but if you have a nuclear war, they have the nuclear bomb, exposure, people with p53 mutation may have much chance to survive than the normal people. So now we know life is a package with all this life insurance. So the humanity actually rely on different mutation or different people. That's why some people is tall, some people is short, some people is overweight, some people is very skinny. They all have its value for the humanity. So the more I'm studying, I cannot simply to say the stress where they come from, but I only know the stress is everywhere coming, including the environment poison, including the chemical exposure, including we just working in the evening, all this stuff could. But on the other hand, cancer also is a disease of probability. They have luckier, unluckier also play these hands as such. That's why some people, heavy smoker, no problem. Someone have beautiful lifestyle, they had a problem. But overall, the lifestyle is complete, to contribute to the different trend. Yeah.
All right. His third question, which he thinks you may have already answered, is, are you saying that further genome chaos also happens in response to treatments after cancer polyploid cells exist?
Exactly. So we actually did the couple of experiments, human and mouse model to see after treatment what's happening. After treatment, the chaotic immediately occur in 48 hours, continue last about 3 weeks, right? And then come down, and then become survival clone always come out. So during this process, the polyploidy, a massive chromosome translocation, everything you can imagine is happening, is dominate. But most of them will disappear, cannot be survived. But the cancer is quite fascinating game. They always sacrifice the majority of themselves to fulfill someone to reach the finish line. That is very striking. So I almost thinking about the information, past information is striking, maybe have some new law there, right? So as you know, Jim described, and many people realize this. So under the stressful condition, the biological system immediately switch to the system which produce change. So they don't care. They don't care the progenitor, the father, the daughter, the son, even the same species. So that's the thing is very striking, right? So they're always different, but they always win. So from that perspective, it's very striking. So we actually did some experiment. You know, my PhD student did this one, but we have for 5-6 years now, we cannot publish them. So what we have done is very simple. We just ask the question, why drug resistance come back when you initially kill more cell? So we just did a simple experiment. You have the cell growing, and then you use the maximum dosage. That's a clinical dosage to kill them. And you use a very moderate to kill them, to what happening. So if you did an experiment in few days, it's obviously the more you kill, you claim the success. But what we are not doing that is we actually to compare the long-term consequence. For example, after a few months, right? Because that's very high. So after a few months, always you initially kill more cell, they come back always quicker, right? So the paradoxical from short-term gain and the long-term gain is opposite. But why is that? We actually realize this because you induce the genome chaos, even though you kill so many cells, and that new genome continue to pass. At a certain point, they become dominant. So we actually ask the student to do the experiment because sometimes you got this result. Sometimes it's opposite, like you kill more, they come back less. So we just did 3 years just for this experiment. We ask a student to see how many times you need to repeat the experiment, which outlier come back. The number is 68 to 100 times. So now, ladies and gentlemen, when you're doing the drug resistance experiment, you need to repeat 100 times, then the outlier always come back. But then we realize this is a game of number. If you initially have so much number, that it should come every time. So then we increase the flask. They come back 1 in 10. And now we know clinically, if you have tumor, you have billions of cells, every time you come back. So now we know that we actually be fooled by the nature. We use a flask doing this experiment. But if you have enough tumor cell, if you use drastically change, they always come back as such. But then people say, oh, okay, someone we cured, right? How to explain to that? I cannot explain that to you, but I only know many people got a lottery, but I dare to tell my son, my daughter, don't study, you will become a lottery winner, right? So that's just impossible, yeah.
What happens to repetitive DNA and genome chaos? James Shapiro would like to know.
Oh, that's a good question. We actually, for many years, we proposed to NIH to study this question. Because what we have found is one of interesting, when we induce genome chaos as a high stress, we always found some of DNA out of the cell as a halo. We suspect those are repetitive sequences, right? So we actually propose, but anyway, so I don't have lucky. I for this many years, I didn't get 1 penny from NIH because of the idea people don't like it. So that's a difficult part for the NIH. But we suspect they have lots of randomness to change because nowadays people found that even under stress, the DNA can easily shuffling. We use lower concentration of lipid staining of live cell, we can say the DNA constantly from this cell jump to another cell is highly dynamic. So it's completely chaotic situation. Yeah.
Paul Davies would like to know, what do your ideas have to say about the BCR-ABL, the Philadelphia chromosome, given that many people have that translocation but do not develop the cancer phenotype?
Sure. So actually, this is a very good question because this BCR, the ABL model is one of the dominant model because people actually have the success of this. Therefore, Cancer Genome Project, everyone cited this one. But we actually found the BCR ABL, the Philadelphia chromosome story is rather exception. First of all, so they only have one or two exception of the cases. The Philadelphia chromosome dominate in the 95% of patients all have such a, the fusion gene. So for most of the cancer don't have such a highly dominate. This is the first. Secondly, we thought the BCR ABL, CML, actually is not really cancer type. This actually belong to the benign type stage, right? So therefore the Gleevec can working on it. Because after few years, they get into black crisis stage. At that stage, additional chromosome changes start to introduce. The drug is no longer useful. Nothing can control it. That actually is a real cancer stage about the CML. So the initially the slowly proliferation stage is not actually typical cancer yet. So that's what we thinking in that way. Also, just for the CML is very let people thinking because some people, normal people also have that fusion gene, but don't have any phenotype. Right, so we know on the surface is chromosome 9 and chromosome 22 translocation, but this is much more story because the coding code changed, not just that fusion gene. Right now, most people focus on the fusion gene, but we focus on the whole genome coding change. So I think in the future people will get more data about this part. I have discussed with Janet Rowley for many times. One of the interesting conversation I remember, before her passed away, is she asked me, she told me, she said, Henry, I also record all this random chromosome change, right? I said, but you did not publish them, right? You record. She said in 45 years, she write down, she observed the slides, she also write down the random chromosome change. I said, you did not report them. And she said, why should I? There's no meaning. I said, if there's no meaning, why you want to record them? I just joking with her. So actually, many people actually noticed that many random chromosome change data. They just did not publish them. They just did not think that useful. If they change their mind, they will have a different picture. Yeah.
Excuse me. May I have a follow-up? Henry, would you please answer the question? The question is very simple. How do you explain people having this translocation and don't have the disease?
Yes, I just told you. Okay. So right now, the genetics actually is very different from previously. Previously, we always thinking if you have this key gene, you have this disease, right? So that's the actually gene way of thinking. So I tell this story is because initially when my mentor cloned cystic fibrosis gene, we always know this is highly penetrated. If you have delta 508, you pretty much get. But then later on, more and more, some people with typical gene mutation don't have the symptom. And now we know they have gene layer of modifier and the layer of the modifiers of modifier, right? So that's why, for the chromosomal change, lots of change, the genetic change is always a potential. They are not really, you have 100% of penetration. In fact, CML is the most powerful. They already have 95%. If you have that one, you have this diseases. But for other cancer, right, is a population behavior, how many you have and at that time. So therefore, for example, if just before the crisis stage for CML, And the patient, some have more severe situation, some have very moderate. So that distribution, so if you ask me this question, I cannot answer why someone don't, but I also cannot answer why some have more severe, some is less severe, but it's the same question. It's a spectrum of from highly diseases, moderate, and no diseases. That's the spectral cost in such a condition. Have I answered your question? Sorry.
Okay, thank you for asking me how did you answer my question. My question, and Dr. Davis's question, is do modifications in the genome cause cancer? This is the question.
Oh, okay, okay. So modification right now since the cancer right now, right? The definition of cancer we have discussed. People talking about overgrowth. Someone talking about the differentiation problem. But we're thinking the cancer is a new system emerge, right? So what to help the new system emerge? We had the so-called the evolutionary mechanism of cancer. Every individual molecular mechanism, if you push enough, they will trigger such balance of system change. So therefore, mutation, oncogene, modification, epigenetic, infection, not happiness, depression, everything actually will contribute to this evolutionary progress. Just in which way and how faster. So that's why so far for people use the oncogene as an example, if you overexpress this one, if you very powerfully push this gene, they will cause to cancer, right? So now we know they have so many ways can push the system unstable. So now we just thinking the idea is no matter what treatment, if you can push the genome extremely unstable, you have the potential to become cancer. But when the system is unstable, they become totally different new system. Most of them cannot make it, even cancer cannot make it. They just evolve, stop, they disappear, right? So therefore, but if you have very unstable, you continue push, you will get cancer. So that's the thing. But even most certain thing you just push someone don't have cancer. So that's the thing. The genome also is unstable. So now we actually into the category of the many of the not black and white field, because we know we did the people's blood check. For most people, the frequency of random chromosome change is lower than 2 to 3%. But for all the cancer patients, is much higher, around 5 to 10%, right? Some people even higher. So we know the quantitative something is going on, but we still need lots of people to take a look at this phenomenon. Yeah.
So I want to make sure, Sisan01, sorry, I don't have your real name in front of me. Did that answer your question? How do you explain carcinogenesis?
No, it does not. I mean, I would like Dr. Heng to explain to us that cancer is not a problem of, let's say, whether the weather is sunny or not, but there is very experimental evidence, very empirical evidence based on theoretical ideas to explain cancer. And I have yet to see whether Dr. Heng tells us which is the theory that he favors in order to collect data to either confirm or discard that theory, which is how supposedly science works.
Okay, let me try again. So the somatic mutation theory always thinking the initial change, the gene mutation and the accumulated few of them, which I disagree. The reason is I disagree is because that is fundamentally, stepwise evolutionary model. So you just gradually change, accumulate a gene, and you become. That's where all the experiment I have to tell you is we actually don't think theoretically that's what happened, right? But we try to define the cancer as a new system emerge. So you may ask, what's the new system? Very simple. We just think a new karyotype emerge, and then become new system. But then if you become new karyotype, but if you did not have massively population, you just sleeping there. So you are not actually clinically harmful cancer, right? So you're just in the dormant stage. So therefore, the gene mutation could help in that stage. That's why after you sequence all the different people, they always have lots of gene mutation. But you will say, okay, yes, the normal people also have lots of gene mutation, but then the mutation, all other mutation, but the behavioral mutation, if the clonal, non-clonal, how the distribution, this become important, right? So that is one of the stories. So for us, we always thinking, like you favor the development, we also thinking development is fundamentally important. If you cell in the differentiation environment, if you become de-differentiation, then you become two systems together. You get interaction, competition, but no matter what happening, for us, the semantic evolution, if you think evolution is involved, you have to have genomics, or you have to have inheritance involved. So that's a key point. So the inheritance right now, they have two big types. One is genetic, genomic inheritance from gene, higher is the chromosome. And the non-genomic inheritance, you have epigenetic change, you have lots of random non-genetic, the physical different type, right? So all of this doesn't matter. As long as your genome fundamentally change, which phenotype differentiate with the normal cell become dominant, then that's a cancer phenotype. That's our definition. So I hope I answered this question. But then the gene mutation, development stages, giant cell, right? For example, giant cell right now, everyone studied, they just all of a sudden, they become many chromosomes inside it, and they also change the surface, have the stem cell feature. But if you study the chromosome, they're all chaotic. And then they just fast dividing, become different population competition with each other. In our sense, they become new system and they become successful. Therefore, they become basis of the cancer. So that's basically we actually try to define.
So Rafi had a comment about that, and then Paul has a question that he asked right before that. But just to clarify, in other words, what you're saying is that the gene mutations are not the primary cause, but rather the symptom of genome level chaos, instability, and reorganization. Is that right?
It's correct, but also later stage, the system hijack different gene mutation to help them to become more powerful. Almost like I joke to my student. So you become king first, then you build the palace, right? Otherwise, you build lots of stuff and it's for other people, not for you. So that is something like this, yeah.
Paul would like to know, do you have any personal thoughts about alternative therapies to deal with cancer, such as diet, reduce simple sugars, reducing stress, meditation, prayer, oxygenation, which is deep breathing and walking?
I think that's a good question because as I mentioned the other day to people, right? So for breast cancer, if you just have someone, your relative take care of you, and the 5-year survival, you can increase easily just 10%, even more. But the individual gene, we never have such a important stuff. So therefore lifestyle, and also somehow to have certain pattern to make the whole individual more constrained, I think is extremely important, because many doctor, physician told me the story. They said someone, everything is fine, the cancer after treatment, everything is good. But then all of a sudden they have family tragedy, someone pass away, boom, all the diseases come back, right? So this is not just one person, quite often. So if you're thinking about this, so then we're actually talking about that is you already have a cancer. It's hiding somewhere with a unique genome, but because of system constraint, you don't have chance. But when this constraint is loose, you immediately, your population become dominated, become clinically, important. So therefore, I think that is is a kind of important the way. So now people, more people talking about if we not to target our own system, but rather than have different constraints. I think that is the similar idea. So we discussed with Bob Gateby to talking about whether or not for the adaptive therapy can further benefit just on the target for the microevolutionary phase. And he said that he's thinking if we know different phase, definitely should be help, right? To thinking about this is one. Secondly, some people use immunotherapy to target particular gene mutation. So if you sequence them, you use that as a profile, you're doing that one. I think that could also be very useful as long as you just control the microevolutionary phase without trigger the fundamentally switch. I think in the cancer, the treatment should muchly improved. So this is from theoretical point of view. Yeah. But again, the other stuff, because to answer Paul's question, there is a mystery in the cancer research field. People actually did not try to really understand what's happening. Is a Ohio group did one experiment. So they have locked the mice, right? After a few months, always majority of them become cancer. What they have done is they just change the cage, right? And they put some of more animal inside there and doing some kind of physical change or whatever. All of a sudden, many animals do not go to cancer. They did not treat anything. They just changed the cage, right? So everyone is discussing this, what's happening? Some people say, oh, maybe introduce stress. Someone said, oh, maybe, it's boost the immune system. But for me, I think what they have done is they just introduce the variable. So therefore the system behavior is changing, right? So most of them do not have cancer, but some mouse may get cancer much quicker. So by triggering all of this circular environment thing, actually we could have something very useful for the drug treatment for the cancer.
Inga Schubert, excuse me. I saw that you had a question, but I saw that you also came off of mute. So would you like to ask your question or would you like me to read it from the chat? Okay, I'm going to read it from the chat. He said, I agree that chromosomal changes may initiate speciation, but if you define that gross rearrangement defines already a new species, then taxonomy becomes difficult. For instance, we have many barley and broad bean lines with homozygous rearrangements, but to call them new species sounds strange to me.
Yes, I mean, I appreciate the way. So the idea use the core genome to define the species is just the beginning. Of course, the community will debate, so where to cross them to draw the line, right? As such, because traditionally people always, to even though use a biological, concept of the species, but then at that time people do not know the detail. Like you are expert to study all these plants, the small change, and how to tell. It's extremely difficult. We have this dilemma ourselves. For example, how about the Down syndrome? They have one piece of chromosome, right? Of course, we say it's a human. We did not say, reclassify this, all of this. So what the question you have is just like, so for this, the subspecies, how to define them. So those actually leave this question to the community. People will gradually figure them out. But now we just conceptually thinking, use the genome similarity as a criteria to classify species. But then, again, they have to be decided by the community, including people like you, to make the decision. Yeah, we just point out the challenge and the possible solution.
That's great. Thank you, Henry. All right. Well, right now we don't have anything in the chat. So anyone who has a question, Dennis has a comment. I think the concept of a species is simply our way of dividing nature up. It is useful, but we should not be wedded to it being clear.
So Dennis, actually previously the people always thinking the concept of species is not absolute, right? Because according to the stepwise change, even for the Ernest Mayr, he's at one hand he said, okay, species is a group of have the sexual barrier and all this stuff. It's correct. But on the other hand, he said fundamentally there is no, the point to separate different species because always the population gradually changes. So therefore you cannot tell the difference. But now we know it's not gradually changing population. We know is that the genome change immediately separate from that individual with another population. So we need to rethinking this one. So I thinking actually the species is become more defined than before under this microevolutionary concept. Because we no longer thinking is gradually change, right? We rather to thinking is drastically changing. For example, even happening in our human population, some individuals, they just have chromosome fusion, right? They become chromosome number become 45. And then because of relative marriage, and they have homozygous of chromosome 44. So I think that 44 immediately departed from our 46 group, right? So therefore is more defined. But you're right, is in reality also has difficulty is some monkey population, they have one chromosome, more, one chromosome less. This is a phenomenon we know. But we do not know the more or less whether or not they actually have disadvantage or advantage of reproduction, and what's the impact for this dynamics as such. But that is a very important question. So that's why we use a core genome concept. That means the majority of them with that genome, but they have slightly vibration, one chromosome more, small piece less, as such. But the overall population actually, is certain, but in the boundary of this hybrid zone, they always have relative vibration. So that maybe, it's some kinds of exception. So we're thinking we need actually modify, but the problem is for bacteria, how to classification, right? Because they don't have. So currently people always use, ribosomal, the similarity 70%, 80%, right? But now we're actually thinking use the genome similarity could answer that question. Just people have to give after compare so much population, just you give a situation because they don't have the constraint. Every time is different. So we did the simulation, the bacteria, they always changing. So now we suspect the bacteria now is totally different from the ancient bacteria in a way. Even though the morphological is still very same, but the genetic information actually constantly changing. Yeah.
Great, thank you, Henry. So anyone who has further questions, feel free to pop them in the chat or take yourself off mute. Feel free. This should feel like a discussion. So.
Yeah. Jim, do you have some question?
I asked my question.
Oh.
In the chat, I wrote, great reply about the price. I hope you write your bestseller, Henry.
So we actually maybe need to team up with some people, to thinking about this, and I discuss with Rafi and all the different people to thinking about, because right now the idea is as soon as we realize the evolution issue, because this is actually very important. Unfortunately, I did not, know Jim and the Dennis, for a long time, because I'm have been very concentrated on in my small world. It's just constantly thinking all the different stuff. But they have, questioned the neo-Darwinian. I mean, it's they publish lots of stuff. Even though I have thinking about this also a long time, but I did not dare to, engage lots of evolutionary biologists, except a few of them, because everyone said that you don't know what you're talking about. So I said, okay, then let me continue study. So with that one, so they have some key questions. So like James, the idea, natural engineer. So all of this is there. So we actually have more information to put this together. So now, actually, I encourage people to review the James the slides to showing the different aspects, like the random mutation, macroevolution, microevolution, all this, the gained inheritance, everything. So we think that actually fundamentally, if you ask me, it's a time to push new framework. Otherwise, it's if not now, when, right? So that's the thing. So I think but I was very surprised because people always said this to me. They said, okay, you be critical to the evolutionary theory, but people talking about this is over 100 years ago. Now you have more knowledge, so you have to be fair. But this is not the point at all, because this is not the knowledge. I suspect, right? I even thinking about if Darwin have all this molecular knowledge, he will be totally confused, right? So that is because that is so confused. So the reason I'm saying this is because only Darwin's view, he can say that how the nature works. But if you get more than molecular knowledge, you cannot say this world is how it works. So that's my point, right? So this is one point. And the second point is I have to mention to some of you who haven't read my book yet, is Mendel. That is also, it's I was shocked when I during the book writing to digging out for this stuff. So his idea is completely based on exceptional data, right? So so he got the green pea, tall and short. He clearly wrote, if the tall one, you have to have over 7 and 8 foot, and the short one, 0.725 to 1.25. If you use taller than this, shorter than that, you cannot say 3-to-1 segregation. So what the knowledge of our genetics is completely based on exception, because only small proportion at the two ends, in the middle, all of this data cannot give you 3-to-1 segregation, right? So that's why nowadays we did the GWAS for the human height. We cannot find because we use normal people. But if we use giant people, use totally development compromised people, we can find the gene. It's a hormone regulation, right? Hormone gene. But for the rest of the people, we cannot find. Is because we did not use the entire spectrum of the data. That's why we propose the fuzzy inheritance is any single gene, the code for the spectrum of phenotype, right? So same thing, when Mendel talk about the green pea, yellow pea, right? It turns out they have the green pea with yellow dots, yellow pea with green dots, and none of them is 3-to-1 segregation. So with this, the Darwinian, neo-Darwinian, based on the Mendelian, all this information, we did a rethinking. But then what to do? I don't know, to be honest, right? I just tell the story and to analyze what is wrong with this data and propose some of the concept people can entertain. But many people feel furiously about this.
May I comment?
Please.
Henry, what I don't have clear is what you are ruling out, because what you are describing, everything counts. So what is that you rule out? Because if everything counts, what's the point of doing research?
That's a very good question. So for the complex science right now, so for example, we even, one of question when I was a student, people always ask, I mean, in the biological seminar, people most ask question is this: oh, you just did the correlation. Someone always say, what is causation, right? But when I gradually, after spending many years in the field, and to appreciate the complexity, sometimes, the causation sometimes becomes a less important question in biological system, right? So as I just said, we actually did this list of how many things can contribute to cancer. So for example, cell cycle regulation, you have a few hundred genes, right? Junction, communication, DNA repair, all of this. So we did a calculation. If you put all this together, the probability is bigger than all the particles in the universe. So then we said, are you kidding? Is that true, right? So it turns out it is true. So the later Don Coffey, who was people always mention, he's actually is a thinker in the field of cancer. He told me, he said, Henry, when I read your stuff, I only read it 3 times. It's the first time I said, it's interesting. Second time, I just tore down your paper. It's impossible. If that's the case, what should we do? And third time, he said, okay, his jaw dropped. He cannot sleep. Because all this, we have how many genes? Each gene you push, you are going to get a cancer in the model system. So that is the thing, how should we do this one? So therefore, we're thinking we should avoid to calculate molecular individual mechanism because too many of them. For example, like aneuploidy, right? People initially always talking about the microtubule. Right now, DNA repair, nutrition, bacterial, everything is linked. So that's actually is a big challenge for us to handle. So I don't know. I don't know. I mean, almost like everything you do. That's why so many papers published, right? Millions of papers in PubMed. Every paper you read, interesting. But if you put all the paper together, doesn't make sense. So that's called the so-called the crisis of the research. So that's why we need a new paradigm, right? So that's why we're thinking, actually, I put more and more thinking about the importance of development in the way, like also the field, how to, to think about this issue. But one suggestion I strongly suggest is because every time you ask, you said, okay, all the cancer cell is the same. Therefore, the cancer is just another layer of regulation. But the problem is they have 2 key experiments. People always mention, like the MIS experiment. What she has done is she put the cell from a teratoma cell into an embryonic environment, then developed a normal cell, right? So this is one. And then later on, people put the melanoma nuclear, put the mice, they claim they have the normal mice. But if you read this paper carefully, they have 2 key information people missed for over 40 years. The first information is when you put the cell from melanoma cell line into the embryonic environment to develop the normal mice, she clearly said in her abstract, apparently you have to have 40 chromosomes. Because if you have different chromosomes, they cannot get out. So that is the key information. So after that, few years later, she asked the same question. Why this cell line, we can put embryonic environment, they can become normal. And she started her cell line. She found her cell line over 95% of the cell is absolutely normal cell chromosome. It's not the cancer cell. right? So she published this one in the Semantic Genetics. I will send some references, you can check them out. And then later on, just 2004, she published a PNAS paper. She's a co-author. And she used the same cell line, cannot generate this. And they did a conclusion. They did a CGH, they found that actually the chromosome have something changed, right? So she clearly writes the statement. Therefore, even though our system is wonderful, everyone thinking it's good, but clearly it's not just epigenetic change. They have to have genomic change. So that's her own paper to talking about this. But that's a paper everyone cites, right? So therefore you ask me, what's the difference? For most of the cancer normal, I just answer is the karyotype always changed. So that's my answer. But how to achieve this change? But the development is fundamentally important because during the early stage, like even Dr. Jinsen Lu, the giant cell, that actually with the full feature of the development, because that is a wrong development force, therefore they change the genome. So we say the giant cell is a phenotype, genome chaos is a genome type, and the produce a new species is the results of the evolution. So that's just what we have, in summarize.
So Dennis wants to know, does that mean that you're ruling out the simple soma mutation theory?
We actually against the semantic theory, because semantic theory, the thing that by the idea of semantic theory, we have to understand, we have to go back to the foundation. The foundation is the cancer is a stepwise evolutionary process. So therefore, your only thing is quickly growing, then you have a chance to have a second mutation, third mutation. But now we're just showing you just reshuffling your genome, you do not need a large population to get a chance for the second mutation, third mutation. So the fundamental theory is incorrect.
And Rafi said, regarding Mendel, fuzzy inheritance equals inheriting a dynamic range of a possible phenotype along a spectrum or region of state space?
Yeah, I think it's a fairly, to summarize that. So therefore, the environment like a scanner, just for the chance. But when you, even though you are tall yourself, but you have this, this inheritance still spectral. So your kids still can be tall or short, depends on another scan of the environment, right? So that's the way, because initially people always thinking, a single gene is you determined, but if you have many genes, you do not determine. Okay, just 10 genes, we have the population so huge, we already covered that, right? 20 genes, we already covered that, but we still don't have, so therefore, it's time to think about the different concept.
So you keep mentioning a new concept, a new way of creating these ideas and understanding what cancer is. If you were to posit what that new basis would be, how would you describe it?
You mean cancer? Yeah, we thinking that, again, we just thinking that cancer is a new system emerge. You have new karyotype. If you become highly heterogeneity and the population become overtaking the original of the system, then you become cancer. I mean, nothing mysterious of that. But another thing, interesting enough, is actually right now more and more cancer actually can jumping from individual to individual now. So that makes people worry, because not just a different animal, is many, just right now just a handful cases, but we anticipate in the future when human population getting old, the cancer become more common. Maybe some cancer will gain the feature of the infection or whatever, as such. So that is something. But the first case already report, one of the surgeon during operation, he cut his finger and he got a tumor, but analyze the tumor karyotype is exactly the same as the patient's tumor. So, it's pretty scary as such. But if you're thinking about the different system, they can cross the boundary, which then it's not totally surprised as such.
So when you say that cancer jumps from person to person, do you mean like cancer becoming infectious?
We are worried about that in the future. Yes, that's what we're talking
Oh dear.
But
I did not know that.
it doesn't hasn't that ruled out with those experiments that were run in the 1970s and 80s, in which people claimed that they had these viral cancers or cancers that cause virus, and then it turns out that that was not the case?
Yes.
So
this,
why are you saying that then we will have cancer as an infectious disease?
No, because if you're thinking about the system, right? So the infection just one feature. So we're talking about new system. The feature is just it's just a matter of time, because right now we have more and more case, and we live with cancer. So lots of things could happen. Otherwise, you cannot explain all the animal kingdom, all these cancer cases, right? So like media.
So, Henry, are you saying that the cancer of the cervix that young women have is contagious? Is that what you are saying?
No, I'm not saying. I'm just worried about in the future we have so many tumor. We're living with this tumor.
No, we are dealing in the present. In the present.
Yeah, present only have
the future. As a biologist, you never have to. You should never tell what is going to happen in the future.
Yeah. So the reason is this. The reason is that we actually thinking the cancer is a new system, new species, cellular species. Therefore, the new species could gain many different features in theory.
Rafi has a question, which is, do you have ideas for how to move forward to discover the rules and principles of genome structure and how it relates to biological function, both normal and disease states? It seems like such a black box at the current moment.
Yes. So currently, people actually did not pay any attention of the genome with diseases, right? So despite the contradiction in the WHO, for the leukemia, diagnostic, they always ask you use karyotype, right? But the people always, so that's the thing. The secondary recent years start to change because the New England Journal of Medicine and the Sequence Genome Project, they all found that for the clinical prediction, the chromosome always better than alien gene data. So that's a start to change. Even people in science publish a paper talking about this. And also, in fact, many people in plant field that did the chromosomal study, like because he's the expert he from Germany. He's studied the chromosome in the 1990. He published many of high quality paper, even surprisingly, even in the Cell or this journal, to talking about the importance of this. But somehow the molecular people just did not carry that interest. So in the plants, people actually did lots of study and how the genome contributed to the spatial aging, everything. So for the mechanistic gene, actually talking about that, like a different way of switch chromosome locally, massively. So this one should, we should convince the study section to funding this type of research, because most people right now doing chromosome is just want to get the gene by using chromosome, but we're thinking this is the chromosome themselves instead of the gene, right? So that's the idea we want to change, but yeah.
Rafi also made a comment that Tasmanian devils have infectious cancer.
For example, and then some of seafood, lots of they have massive leukemia and everything. So people gradually understand more as such. Yeah.
Wow. That's news to me. Well, what a thing to think about. Well, we've only got 5 minutes left here, everybody. So if you have a burning question, your time to ask it is running short. So please pop yourself off of mute and ask any final questions.
Just a comment. The Tasmanian tumors then the Tasmanian devil's tumor is a metastasis that is going on, that is being transmitted sexually. It's not a new tumor.
Yes, yes. But then where does it come from, right? So always have before that. Yeah.
Via bites to the face, according to Rafi.
Yeah, that's one of this, right? So that could be, because initially we're thinking that the cancer is just out of control growth, but when we realize it's a new system, is a new species, cellular species, then it's pretty scary if you're thinking about it. But from the, I will ask Dennis one question to you advice about is, we actually found something quite interesting or fascinating is, so during we put the stress for the cancer, right? So the cancer desperately change, but most of the changes are going to dead, but they always win this game. So it's fascinating to see how the information survive. You know, have you ever thought about how the information survive by creating new, just as a way of survive the information? I mean, I'm not talking about the specific something, just the information themselves survive. Is anyone discuss this type of phenomena?
Was that a question to me, Henry?
Yes, it was.
Yes, okay. Well, I mean, the standard evolutionary biologists would simply say they have to replicate. I mean, this is why they put so much emphasis on the replicator vehicle concept. But that's just giving the standard modern synthesis idea. As you know, well, I'm not at all enamored of that particular synthesis. So I'm not quite sure what else to say over and above that. But that's what they would say. You have to have replication. That's, of course, why they put so much emphasis on evolution being essentially change in the genome.
So the thing is, you know, because it's not simply replicate, right? So they just they actually survive by creating new information. So therefore, so those current, you know, the organism is totally selfish.
Well, I have, well, this will go on too long, Henry. Within 3 minutes of us being told we're finished. I have great problems with the selfish gene, as you know.
Yes.
That's for another time, isn't it?
That is the wrong question to ask to Dennis.
Okay. Anyway, yeah.
Absolutely, Carlos.
We appreciate you chiming in, Dennis. I know it's pretty late over by where you are.
Okay,
so
thanks.
It's well past my bedtime.
Yeah, thank you.
Yes.
Well, we've got one more question, and then we're going to have to cut it off here. Paul would like to know, how does the idea of chaos match with Shapiro's ideas about the cell's own intelligent reengineering / restructuring? Maybe these two ideas go together, and maybe they depend on conditions.
Yes, I think we actually pretty much on the same way of thinking. So because we can think, you know, the genome chaos is powerful example of how to re-engineering this process. So we actually thinking the genome chaos is a process that involves lots of random steps or agent, like the material is different, stress is different, and the products of a range is different, but the fundamental evolutionary consequence is the same, is create the new information. So from that one, we actually share lots of ideas. So we may more closely working in the future to, you know, to push this concept. Yeah, he's a great guy. So there are a lot of things for me to learn, and then we can push forward with Dennis, with Jim. So that's you know, beginning. The cancer is a important vehicle, but now after studying cancer for so long, I'm more interested in for the evolutionary and the genetics theory. as we're speaking.
Thank you so much.
Yes,
Thank you.
thank you. Thank you so much. And Paul, thank you for your
Henry.
excellent
Thanks.
questions.
Hi, Sandy.
Henry, I really think all of you should pair up and become like a scientific cancer fighting Avengers, honestly.
Yeah.
I think. Carlos says no.
That did not convince Carlo, but I will send some information to you. Back me what I'm saying. Okay.
Thank you very much. I will appreciate it.
Yeah.
Thank you all for joining. I hope to see you in our
Bye,
future
Bye, Dennis.
Q&A session.
you all.
Thank you all so much.
Yeah. Okay.